application of data-driven approaches to flow cytometric analysis of basophil activation testing together with Dr. Sarita Patil. My name is Michele Romano. I'm a product manager in Buhlmann Laboratories AG, responsible for managing Flowcast, and I'm available if you should have further questions. In Buhlmann are more than 20 years that we have launched our first version of basophil activation test, and today we are proud to be the unique company in the market offering a full platform for that testing, comprising our lead product, the Flowcast, and more than 150 allergens specifically developed and standardized for the use with our assay. We have focused on any aspect of the standardization of a functional assay based on basophil activation, including the analysis of data generated by flow cytometry. That's why we are encouraging and supporting research in these fields. Today, I'd like to introduce to you Dr. Sarita Patil. She is currently an instructor at Harvard Medical School, which has focused on the humoral response in food allergy immune therapy. Her long-standing interest from her postdoctoral training in computational approaches for scientific reproducibility has led to development of a pipeline for flow cytometry basophil activation assay analysis. A laboratory will focus on the humoral immune responses in allergic diseases and the modulation of effector cell responses, including basophil activation. In this webinar, Dr. Patil is presenting her efforts to develop a novel data-driven flow cytometric platform for the analysis of clinical basophil activation testing, providing a high throughput, objective approach to basophil activation analysis. Before giving the word to Dr. Patil, if you have any question during the webinar, please you can write in the chat and we can discuss it just at the end of the presentation. Thank you.
Thank you so much for inviting me to discuss our efforts on the long-standing project of ours, namely this application of a data-driven programmatic approach to the analysis of flow cytometry data generated from basophil activation testing. And I really began these efforts during my postdoctoral fellowship, as Michele mentioned, in Dr. Wayne Stressler's laboratory, which I hope to continue on my own. So basophil activation testing has really wide applicability to the field of allergy. And basophils are this very rare circulating allergic effector cell whose high affinity Ig receptor binds to surface IgE. And then this Ig is cross-linked by allergen. Basophil degranulation can release pro-inflammatory mediators, including histamine. Surface CD63 upregulation of basophils has been shown to be associated with this histamine release and is a marker of basophil activation as discovered by Dr. Edward Knoll. And as seen on the flow plot, there is a bimodal distribution of CD63 on activation of basophils. Basophil activation has been studied in many allergic diseases, including environmental or inhalant allergies, as well as food, drug, and venom allergies. It's also been studied in food allergy, both in diagnosis as well as clinical trials of allergen immunotherapy. Notably, basophil activation testing really involves ex vivo allergen stimulation of peripheral blood basophils. And the assay for the upregulation of this activation markers, data acquisition is performed by flow cytometry. There's so many challenges to basophil activation testing and several key sources of variability in basophil activation testing. There's biological, technical, and analytical variability. Technical assay variability has been largely addressed by the development of rigorous protocol standardization, including allergen standardization. And our work today that we're going to present really utilize basophil activation assay using this commercially available kit that standardizes allergens as well as a technical protocol. Another key source of variability arises from challenges in analysis. And analyzing this very rare population of cells, using flow cytometry is very labor intensive and a little permissive. And then furthermore, generation of an antigen stimulated dose response curve and being able to analyze that reproducibly across samples poses a fairly large source of variability that we sought to address using this computational approach. And we were hoping that by addressing this, largely using the approach that we would be describing, we would then be left with biological variability so that we could capture that and more effectively study it. And so the gold standard right now for flow cytometry data analysis is really to use manual gating, often using visualization of cell clusters. And as you can imagine, this is a bit subjective and it can be permissive. It's extremely labor intensive and requires a good deal of expertise in the ability to gate on those cells. On the other hand, we were hoping that by automating this approach, using this what we called auto gating approach, this computational approach, we're trying to produce a reproducible, transparent way of gating on the cellular assay that would then allow us to not only promote that transparency, but also allow it to be applied in a high throughput way at multiple centers for clinical applicability, and also to sort of highlight the ability of this program to then capture quality control measures that should also be part of the analysis and transparency. So the clinical data set that we used in the publication that we recently published on this approach was focused on peanut oral immunotherapy. It was a single center trial of peanut oral immunotherapy with 30 pediatric patients. So we had very small amounts of blood, as you can imagine. These patients were peanut allergic by skin prick testing with specific IgE, and were then underwent a fairly standard high dose oral immunotherapy protocol where patients get a modified rush followed by a build-up period, a maintenance period, and then an oral food challenge to determine desensitization. This protocol actually also had an additional arm to study sustained unresponsiveness by having patients avoid peanut for a month and then re-challenging them to look at how many patients continue to have long-term tolerance. The Basophil activation testing component was performed on peripheral blood that was collected throughout this year-long protocol. And so we have baseline blood, but then also blood on an almost monthly basis during the build-up period and then during the maintenance period once and during the food challenge period. And so even though the study is fairly small with only 30 participants, it's a very longitudinal study that captures many, many data points for each individual patient. The basophil activation testing was performed on peripheral basophils by stimulating 50 microliters of whole blood with various peanut antigens, so ARH1 to 6 and whole peanut extracts, for using that standardized protocol from the Buhlmann full cast kit. And then after stimulation, these basophil data was acquired by flow cytometry. It should be noted that flow cytometry captured 2 fluorescent markers, CCR3 and CD63, as well as side scatter characteristics. This data was then analyzed and could undergo quality control statistical analysis after that. And the analytical component of it includes traditional components of analysis, including compensation and transformation of flow data, as well as removal of boundary events. Quality control, where measures that we thought would help us identify outliers or experimental flaws. And then statistical analysis on that cleaned up data included looking at dose response curves and area under the curve. So we took a very stepwise approach to the development of this algorithm or programmatic approach. We first, out of the entire pool of 294 experiments, each of which had at least 25 experimental conditions, we took 52 of those for the development of this programmatic approach. And this meant that we kind of reiteratively used that data to develop a stepwise gating and what we thought would be a robust and reproducible program. And so the program itself was written in an open source platform based on R and bioconductor packages. The bioconductor package that we use is Flowcore, and that Flowcore package provides a very strong structure for the flow cytometry data. That is that it uses the flow cytometry data acquired for each experiment, treats it like a flow set. For each SCS file, is loaded in as a flow frame. And pheno data is the name that they use for the metadata that sits above all of those unified SCF cells that's captured in every single SCF cell. And by separating out those two characteristics, then we can analyze the data and then also be able to analyze it not only from a single experiment, but also from multiple experiments. All of the code that we use for developing this package is freely available on my GitHub at the following address. So there are two main functions that we develop part of the programmatic application. The first function is a BAT gate, and that function was really designed to identify those basophils and then subsequently identify activated basophils, or the percent CD63 upregulated basophils. We use a very stepwise gating strategy that I'll go through in a little bit. That stepwise gating strategy really involves first, again, removing any boundary events and transforming the data. And these are standard approaches that are used in flow cytometry gating by other software often used in manual gating, for example, like FlowJo that we used for manual gating in this particular paper. We then also selected size scatter low cells. And here, the distribution of the size scatter low cells either fell into a distribution that had two groups, in which case we called it k-means of 2, or three groups, in case we called it k-means of 3. And you have to be able to input this data into the function itself, and that's developed to give the function a little bit more flexibility depending on the flow cytometry characteristics of the population that you set, or the PMT gates and the acquisition of the data that you set. Selection of CCR3 positive cells was subsequently done, and then we finally gated on that basophil population by looking at the distribution of CD63 and gating on what we thought were activated basophils. That stepwise approach we thought was very comparable to a manual approach. So in the top, we see the manual approach where you gate on singlet cells, and then you look at the side scatter low CCR3 population that's identified as basophils. And again, you know, how you identify that population of basophils or how you set that gate when you're doing it by hand is a bit subjective, whether you constrain it to a smaller circle than is seen or a larger circle than is seen is really highly dependent on the operator. In auto gating, we focus in on side scatter low cells by iteratively gating. So again, this is where it matters, where there's, in this case, there's 3 k-means populations here. So we gate on side scatter low cells. And then we gate again on CCR3 high side scatter low cells. And then we gate using a k-means of 2 to kind of decide on the most robust and reproducible population of basophils. And that really means that their side scatter and their CCR3 distribution is normally distributed in 2 dimensions. That's what the k-means of 2 really means. So like I mentioned before, you have to give it the BAT gate a couple of pieces of input. One is where the working directory that you have, the R, or where all your FCS files are actually residing, so that it knows where to import those files from. The fluorophores refers to, you know, whichever fluorophores you're using on your machine to identify CCR3 and CD63. Side scatter number, again, this refers to whether or not you have a distribution of 2 or 3 on your flow cytometry acquisition. And this is really determined by how you set your PMTs for side scatter. And then finally, the med control tube that tells us where the resting basophil tube is, so that we can use that for gating, that we'll get to subsequently. The outputs of this function are a CSV file that have data statistics for each stimulation condition, and these data statistics are focused on the activated basophil population. So it tells you the percentage of CD63 high basophil. It can also give you characteristics of fluorescence for the entire basophil population. So characteristics such as CCR3 MF5, CD63 MF5, the covariant variation for these different fluorophores, etc. And then finally, it gives you a PDF file of the gating graphics for each of the conditions, just so you can visualize whether or not the gating is accurate across all these different conditions. And this was really essential in the development of the program, because it allowed us to go back and kind of verify that our program was indeed gating on the population of basophils that it should be gating. The next function is the BAT measures function. This is really designed to derive a dose response measure for antigen-specific basophils. And there are two main measures that we have this function derive. The area under the curve measure, which is just derived using the log of the stimulation concentration. And again, we'll get to what that actually means. And the ED50 of the dose response curve as well. The input for this function is data from the previous BAT gate measure. You can actually use this function without having used the BAT gate function if you have data in the correct format. And then that data essentially needs to have grouping variables to identify each dose response series. So for example, if you have several different patients and you're looking at, one antigen for several different patients, you just have patient. In this case, because we had a number of different antigens. We had patients, we had the longitudinal visit means, and then we also had the different stimulation conditions listed. And then the output file is just the name that you want to give the file that that will be then the CSV file that has all of the different characteristics like ED50 and AUC of those response curves. So once we took the algorithm that we developed with those 52 randomly selected experiments, we then applied it to the entire population of 294 experiments, and we're able to get a 92% success rate for good quality gating of basophil experiments. And so this means that we gated using this algorithmic approach more than 7,000 individual BAT conditions. So one of the questions that we thought it would be very interesting to answer using this programmatic approach was to answer the question, what is the right strategy for identifying these bimodally upregulated, activated basophils? And the difficulty was that there were two different approaches that one could imagine you would take to this. When you have a bimodal upregulation of CD63, those activated basophils could be either identified by setting the gate here, which is between the CD63 negative and CD63 positive basophils, or you could set the gate on resting basophils by saying, oh, well, we think that the CD63 gate should be somewhere between 2 to 2.5% of that CD63 distribution on those resting basophils. And the difference in these two different ways of identifying the CD63 gate looks fairly subtle. If you look on an individual experimental basis here, you can see the cutoff is a little bit to the left using the resting basophils and a little bit to the right using anti-IgE-based basophils. But we thought, let's compare this to hand gating, because when we do this using a programmatic approach, we feed to the program, okay, the anti-IgE stimulated basophils we know are going to have a bimodal distribution, so they should have a k-means of 2. Find me the midpoint between those two different peaks, and that's how we would very rigorously select this. When you're doing this by eye, of course, you're just picking what you think is in between these two different distributions or these two different groups of basophils. So we compared medium-based gating using the program to medium-based gating using manual gating. And then we compared anti-IgE-based programmatic gating to anti-IgE-based hand gating to see which way is the most robust way of setting the CD63 gate. And it turns out when we compare how well we do across these different conditions, that resting basophils actually have a higher coefficient of linearity. So they have 0.96, and that's a significant difference between this type of gating on resting basophils versus anti-IgE activated basophils. And both of these graphs, the manually gated basophils are on the x-axis, and the auto-gating basophils are on the y-axis. And you can see they just clump better in the resting basophil condition. So we thought that was actually very interesting. It was kind of unexpected and not what we had predicted. And certainly very valuable, I think, to the application of basophil activation testing across a wide variety of allergic diseases. The next question that we had was, how do we robustly be able to analyze this complicated dose response curve over time, longitudinally in these patients that are undergoing an intervention? And so in this particular panel, we're looking at a particular patient basophil activation testing across different allergens and across different time points over the course of OIT. And this top set of panels is manual gating or hand gating, which is the gold standard, and the bottom panel is auto gating. And it's very easy to see if you just compare the two that they both look very similar, which is reassuring, of course. But, you know, we also can already see that this might be complicated to analyze from a dose response perspective, because while you have very beautiful dose response curves, at baseline, these curves start to flatten out. And if we look at the different patterns of what happens over time in these OIT patients, we see different types of patterns. So again, these panels are lined up in time on OIT across the top, and different allergens across top to bottom. And again, for each individual panel where there's a log of antigen concentration, the percent CD63 high basophils on the y-axis, we can see that these beautiful dose response curves that occur early during OIT really start to change in configuration over the course. And how do we capture how this patient looks from, say, this patient whose curves completely flatten out? And so there are different ways of analyzing this dose response curve, and two of the main measures that have been studied involve the CD sense or ED50, which is really taking 50% activation from maximal activation during that dose response curve and looking at the antigen dose that induces that. CD max, or the maximal amount of degranulation that is seen in a complete dose response curve, has also been used as a measure of BAT. And, you know, in one of the reviews that Wayne Scheffler and I wrote together, we thought, well, gee, maybe a more reproducible way of capturing another measure of basal activation might be using area under the curve, where we use a log of antigen on the x-axis, the percent CD63 high basophils on the y-axis, and we just sort of look at the trapezoidal area under the curve. So this is really data-driven, because you're not imputing values here, and you're just being able to kind of look at the area and compare that over time. This measure has its downfalls as well, because while it can discriminate between conditions like this and this, where you can see less area under the curve here and a greater area of the curve here, which is kind of consistent with what you might think when you're just kind of looking at these different dose response curves, it's not perfect, because it does weigh the higher end of the antigen concentration a little more heavily than the lower end. And so I think comparison across the same data set is in some ways a little more simple than across different data sets. When we started applying these different basophil measures across our entire data set, we began to find that it was a little more problematic, because as I described previously, we have a lot of these dose response curves that flatten out tremendously, and where you're not actually seeing that entire sigmoidal dose response curve that you're used to seeing, that is used to derive an ED50 or say a CD max. And so what we actually did, with the help of a very talented biostatistician named Augustine Calatroni from Roe, was we were able to test a bunch of different ways of developing a model. So we used a mean, median, mean square models to kind of derive dose response curves across each one of these conditions. And then using that data, we're able to select what the best dose response curve was based on characteristics of how well it fit the curve, and then derive the ED50. And so that's really been incorporated into our BAT measures function that allows us to so robustly be able to capture that ED50, regardless of the variation of that dose response curve that might occur during an interventional trial, for example. We learned a lot of interesting lessons from quality control measures as well. So when I was first doing these, starting to develop this program, one of the first quality control measures I thought that was an obvious quality control measure was to capture the number of basophils that we were seeing per each stimulation condition across these experiments. So on the x-axis here is the acquisition date for every single individual basophil activation experiment, and the y-axis is either basophil count or CCR3 MFI. I'm sorry, that's a typo. And so when you look here, you can see the basophil counts actually vary quite widely. And we were, of course, concerned about those experiments where there were lower basophil counts, whether that was because of a difficulty with a specimen or difficulty based on time for acquisition. the interesting thing, of course, was that all of a sudden we saw this very interesting group of experiments with incredibly low CCR3 MFI. Now, it turned out that these experiments were all done on Christmas Eve one year, where there was probably a great deal of technical variability due to unexperienced operators doing this particular set of experiments. So I think that this kind of shows the value of doing quality control measures across time, especially when you have a set of clinical data that spans 5 years like we do. Another really interesting question that I think we were able to address to our satisfaction was, what is the minimal number of basophils that you need to have to be able to derive robust measures of basophil activation? So the obvious measure of basophil activation that we use is the percent CD63 high basophils. And so using our entire compendium of thousands of tubes, or experimental conditions, I should say, we decided to say, what if we bootstrap those samples and sub-select different quantities of basophils? So on the x-axis, we have we start out with very, very low numbers of basophils, so 50, starting at a 50, and then go to 100, 150, 200, et cetera, all the way up to 1,000. On the y-axis is the difference from what we derive if we use those small populations of basophils from what the tube's actual value was. And so when we do that, we find that, well, obviously, if you use very, very low numbers of basophils, like 50, you're getting huge amounts of variance. And this is absolute variance, by the way, and not just the actual number, which is why it's all positive. As you increase basophil sampling, that variance really decreases. And so, when we look at this data, you could choose to set the minimum number of basophils to be whatever number that you're most comfortable with. But I think that this is valuable information about what that number should look like. And for us, we felt most comfortable around 700, 800 basophils. We can see that difference is really falling, say, less than 1.51% of variability. We also wanted to see whether this downsampling could inform how different area under the curve measures might be of these dose response curves. So we now bootstrapped again, varying numbers of basophils located on this x-axis. And on the y-axis, we looked at the covariant variation of the area under the curve if we were to derive it again using our trapezoidal method that we had established in that BAT measures function. And when we do that, you can again see 50 basophils gives you a good deal of variance here. But as you start to decrease, as you start to increase the number of basophils, you decrease that variance considerably. And so again, for an assay, to decide that you wanted a good margin and clinical reproducibility, you could certainly set that you wanted that covariant variance to be less than, say, 0.1%, and could see that you need about 600, 700 basophils to achieve that. So this program that we have, recently applied and published is really just the beginning. We really wanted to create an open access method that could be applied widely. When we applied this programmatic method, we used a cluster because the computing needs of course, analyzing the almost 300 days of covariance that we did required quite a bit of computational power. And so, this program has been created so that it could be used on a cloud-based system. And the idea is that this program should be able to be applied to multi-center data. We've already shown that it's robustly applied to data that's acquired longitudinally across many different years. We also think that this approach can be used to set standards for analysis of flow cytometry gating of basophil activation testing. I think the future of this program may include machine learning approaches where the program can be taught to robustly or reiteratively gate on basophils in a way that can adapt to different instrumentation and different conditions and even different patient populations. The quality control measures that you can analyze using this approach, I think, are particularly powerful because for the first time we're able to really look at the impact of the quality of basophil activation in these large longitudinal studies, as well as for internal facilities to kind of look at their quality control and even control for any technical variation that might be present. There are many different applications for this approach. You know, some of the other obvious applications include using this with other allergens, or to use it with multi-marker basophil experiments. And we have already started applying it to other data sets that have multiple fluorescent markers for basophils. And so I think that is definitely something that this approach should be applied to. And I'd like to acknowledge this huge team of people who really helped make this huge project possible. So the person that I have to that I collaborated very closely with and owe a great deal of debt to is Augustine Calatroni from Roe, who did all the biostatistical work to help derive those measures of basophil activation that I think really add so much value to this data set. And Michael Schneider from Buhlmann Laboratories was very critical to the development of this program 5 or 6 years ago when we first thought about it for the first time. I would add gratitude to my mentor, Wayne Trepler, who encouraged me to go down this road to begin with, and our many, many technicians who over the years have done the basophil activation testing in our laboratory to generate this large data set. Finally, the clinical data was impossible to generate without our clinical research team, our food allergy research team, the Harvard Catalyst, which provides a tremendous amount of clinical resources, and the patients and families who've donated endless amounts of time to this effort. And then finally, I have to thank the people that have funded me, including the QuadAI and FAIR, as well as the NIH NIAID. I'd be happy to take any questions.
Okay, thank you, Sarita. Now I'm just checking for, and I'm opening a webinar to a question and answer. I'm just checking for some of the questions. One of the main questions is that which are the, you have talked in one of your last slide about the future of the program. Is this, what are the plan, the concrete plan, how the assay could be further developed? Do you think that you have a 2.0 version, more user-friendly, or something like this?
Yeah, no, that's actually a really great question, because right now, you know, application of this kind of approach is kind of limited by, you know, the need to use these tools, R and bioconductor, and kind of understanding, having a rudimentary knowledge of programming to be able to apply these, and also to be able to use, like, for example, Linux-based cluster. And so, yeah, no, there actually are a couple of very useful ways of applying a more user-friendly platform. So one of the ways that people have implemented for other similar programmatic approaches for other large biological data sets include using something like Shiny, which essentially puts a web-based HTML page where you can feed those characteristics that you'd otherwise have to program into a script. Now you can actually just feed those characteristics into a web page that asks you kind of direct questions, so it's not so confusing, doesn't have so much programming lingo. And then that can also be fed directly into a cloud-based server, for example, like Amazon Web-based services that is behind a firewall and that, you know, can be used for clinical data analysis, and can definitely carry the computational burden of analyzing these many experiments. So I think those are some straightforward ways of kind of making it really, really applicable. I should go into a little bit more of the processing power that I needed for the application of this algorithm, because it's not something that I often talk about, but I think it's important to know. So when I was in the development stage, I just used a single core approach, but like in the application stage, you know, I had to actually use a high performance Linux-based computing cluster. So we had an institutional cluster, and I transferred data in its compressed form, then uncompressed it, and then used parallel processing of individual experiments using a wrapper script for the function that I've already provided. These are very technical details. It doesn't actually have to be this complicated. I think in the future, this can all be moved to a platform from sort of like an Amazon instance that can be launched by many different users for their own purposes on their own separate individual firewalled clinical data sets. So that's one way of doing it. Using a Shiny web-based server is another way of doing it. Amazon has certainly hosted these sorts of applications before, so that's not a problem. And that's only one route. There's many other places where cloud-based servers have provided computing power for these kinds of approaches. And then finally, the data cleaning and visual inspection that I did was really local, and the statistical analysis could all be done on one computer.
Okay, I have just some more questions that are coming in. One is quite easy. How easy is to implement in routine allergy diagnostic department?
That's a really good question, and actually that's the reason that I even started on this path in the first place, was that my own department, the head of our immunology laboratory core, told me that, you know, he thought that basophil activation was going to be very challenging to implement locally for our facility because of the difficulty in analysis. So the whole point of developing this in many ways has been to make some of that more standardized. Because you can basically take this and implement it locally without the need to use any additional machines or any additional resources, basically makes it completely free and makes it a lot easier to implement. But you would still need, I think, some infrastructure and some support. So you would definitely need a way, you know, the elements that people still need today for flow cytometry analysis include sort of a place to put it that's firewalled and secured, and the power to analyze it. You can get away with analyzing basophil activation testing from one center using one computer if you so choose. You don't have to do it the way that I did it, but our throughput was so large that I had to use a computing cluster. Does that make sense? Did I answer the question?
Yes, I think yes. I've just two quick questions that are technical questions. One is, can the analysis be done by markers independent of CCR3, for instance, CD203C, CD123, including HLA-DR? That's the first technical question.
Yeah, no, of course. And that was actually what I was alluding to when I was talking about the broadening the application. So I've actually been working on an approach for multi-marker or multi-color basophil experiments using those exact markers. So in the case that in the data set that I'm using, which is 200 plus experiments, but they include CD203C, CD63, CD123, HLA-DR, and CRTH2, all as markers of basophils. And so I think this approach is perfectly feasible for that too. We haven't actually published that yet, or it hasn't been QC'd enough for me to put it up, but I hope that version 2.0 of the program that I have up on GitHub will include that.
Okay, I think that there is time for another two questions. One is quite, again, technical, and another more general. The one is, we have started with bath with penicillin antibiotics. How usable is your method for penicillin, beta-lactams, and other drugs? If you have any experience.
Yeah, no, I think that's a great question. I do think that because we found that CD63 gating on resting basophils is a more robust way of identifying activated basophils, that really helps broaden applicability of this particular approach, because it doesn't require that someone necessarily have reactive basophils to one dose of anti-IgE. But I can't speak to penicillin directly, as I haven't actually analyzed that data myself. But I would expect it to be useful. Yeah.
Okay, and just the last question is, what about no, the quality criteria is okay. Again, you have already spoken about the 600 basophils. How far we are from the implementation of the program in the clinical laboratory implementation? I see. I can imagine that the question is about the standardization in CRO labs, in clear recommendation and stuff like that.
Yeah, to my knowledge, this program has not been used in a CLIA certified lab to do Basophil activation analysis. But I was hoping that by providing the this program freely, that would be something somewhere that this program would be useful, or most useful actually, so that you could actually do multi-center Basophil activation testing reproducibly and get the same results every single time using standardized, technical protocol, but then also standardized analytical protocol.
Okay, so Sarita, thank you very much and for your talk. And I would close the webinar. Thank you to all the audience, and I would just to remember you that we will upload on our website a recorded version of this webinar. So thank you very much and have a nice day.